We'll start now. Am I audible?
Yes, yes.
Hello.
Yes, you are.
Yes, yes.
Okay. Good morning, everyone. Welcome to the first talk of the Dialogue with Destiny series on the topic Insilico designing of developing derivatives of targeted inhibition of cancer cell signaling pathways. I, Rosalind Swamy, will be acting as the moderator for this event today. This series is a flagship event conducted by the Bioscience Society NIT Rourkela, which is scholarly registered under SAC NITR. For the very first talk of this series today, we have invited Honorable Speaker Dr. K. Venkateswara Swamy, Associate Professor at MIT ATT University, School of Bioengineering and Biosciences. Next. Next slide. The pandemic has really damped the spirits of research and innovation among both the students and researchers. So, to fuel us up and inspire us, we have created this series where we reward the professors from all across the globe with diverse field with expertise will be called upon to share with us their insights into the field. Well, this is the very first talk of this series we will be listening today. Next. Now let's move on the main agenda. As I have already mentioned in the beginning, our honorable speaker for this talk, Dr. K. Venkateswara Swamy, he is an associate professor from School of Bioengineering Sciences and Research. Dr. Swamy has completed his PhD from Sri Krishna University, Andhra Pradesh, and his thesis project was computer-aided drug design studies on human histamine A2 receptor. He was on Officer Lab Administration at Thyroid Technologies Limited, Navi, Mumbai, India. He was the Associate Professor, Dr. D.Y. Patil Biotechnology in Bioengineering Institute, Pitapik, Pune. He took up the charge Associate Professor at MIT School of Bioengineering Sciences and Research MIT Art Design and Technology University, Pune. He was awarded with Young Scientist Award by ICN PhD Judge, Washington, USA on September 8, 2012. Also, he was a visiting faculty at Jaffa University, Sri Lanka. He was IPSC coordinator as well as NAC coordinator. He was also a reviewer of various international journals like Journal of Molecular Modeling, Journal of Molecular Modeling and Graphics, Journal of Biomolecular Structure and Dynamics, etc. He had 40 publications and did 3 government-funded research projects and 6 joint research projects collaboration. Next. Now it's going to be very much exciting and each one of us shall learn many insightful concepts on computational biology. So without wasting any time, I shall now request Dr. Swamy to deliver a talk. Before the talk, I would request everyone to please mute yourself during the talk. If you have any questions or doubt regarding the talk, just write it down in the chat box and we will be noting it here. After the talk, your question will be answered. Now I would request Dr. Swamy to start his talk. Sir, please. Hello.
Yes. Good morning. Thanks for nice introduction, Rajaliani. And I'm happy to share my research work, which has been conducted in our lab. So I'm going to present the research work based on the cancer drug targets. And basically, these cancer drug targets are existing in the human body, and we will see that all the topics in my presentation today. Okay, please confirm that is it visible to all of you?
Yes, sir, it's visible.
Okay, fine. So here, my research topic here is Insilico designing of degulin derivatives for targeted inhibition of cancer cell signaling pathways. So if you see the title is very big, but just I would like to make some distribution here. So number one is that the word is in silico is nothing but whatever designed is on computer based. Nothing but we have taken the data from the laboratory information. Then we have designed some of the drugs, or I can say that some of the compounds which are very essential to treat the cancer. So we have designed based upon some of the parameters. Then those compounds were tested in computer. So these targets basically for cancer cell signaling pathways. So we will see that what type of pathways are there and what are the drug targets. Now can we look into these cancer types? When you say the cancer, so it can able to spread throughout our body. When you start here in the male and the female, little bit of a difference in the type of a cancer when as compared here the image which is given by the World Health Organization, you can see that there are maximum cancer types in female as compared to the male. In the same way that when we look about that estimated number of new cases may rise or already existing in 2020, so we already completed our 2020, but as per the cases which may be mentioned by the WHO, so it is showing almost the breast cancer cases almost all 24.5%. And when you see that the lung cancer may be 8.4% here, but other cancers you can see that 37.8%. And remaining cancers already mentioned, among them, one of them are thyroid cancer. It is 4.9%. So we may be considered as a 9% of throat, or it may be thyroid cancers may be considered in this case. So we can see that the throat cancer, cancer of the esophagus, lung cancer, breast cancer, stomach, bowel, cancer of the bladder, and then cancer of uterus, and then skin cancer. And one of them prominent right now, you can see that the ovarian cancer. So here we can find that these many cancers, but there may be one region or the one uh, target may be there, nothing but with the protein or the enzyme which may be involving all the cancers. So this type of thing can be extracted or identified with the various studies. So here we can see that the Professor Raghava has been done a cancer DR database on all the cancer drug resistant database as it is. So even you can see that the targets when you consider into this image which has been published here in the 13th March 2013. So here we can see the lung cancer targets are more when compared to the other cancers. So here we can see the 185 drug targets are there, and then liver related with this, so 19 drug targets, and then kidneys maybe 26 drug targets. Nothing but these are the proteins or the hormones or the DNA. So among them, we can see the maximum scope when you look up at the targets to treat currently the lung cancer. But when you look into that other type of a cancers, they are a little bit of less as a drug targets. So now the companies and as well as some of the industries are working into this to target some of the proteins where you can stop their biological action, which may be influencing or enhancing the cancer type of a disease. Now, whenever we have the disease, alternatively we have to search for the treatment. So when you look about the currently available treatment or immunotherapy, radiation therapy, targeted therapy, chemotherapy, again bone marrow transplantation, surgery, and the hormone therapy. So when you look into this, a necklace of a treatment, in this case, most of the time we may be looking about the chemotherapy, nothing but we are going to give some drugs to the patient so that we can try to treat the cancer. If it is not possible, then we are going ahead with the surgical methods where you can remove the complete tumor from the patient. So it may be from any part of the body. So even we have the history of so many uh, people that how they have come up out of from the cancer disease. Now, when you look about that, this is see the publication, or it may be that the Chennai almost 90% cut in the price of 9 anti-cancer drugs. So here the Chennai government has been taken a decision that we have to reduce the cost of these cancer drugs because it is very high to purchase these cancer drugs by normal people. So here we can see that the generic name, nothing but the main name of a drug mentioned about that Yellow tibinib is the 100 mg tablet is almost the pack size 10 tablets going. Previous rate is 6,600. So now it has come down completely 1,840 rupees. So like this now everybody looking about the decrease in the price of the cancer drugs from higher level to lower level so that can be able to reach the poor people of our Indian country. Now further, when look about the market, exactly the pharmaceutical industry, how they are growing these cancer drugs by selling from their point of view. So here we can see that the Allied Market Research has a global oncology cancer drugs showing that it is 2,22,380 million dollars price may be going to high in case of just by selling the drug cancer drugs. So it means say that we are going to enhance this cancer disease maximum or proportionately it also increase the price in case of drugs. So it is not feasible to common man to get treatment with the drugs which are very high in range. So in this case, now it become as compulsory to know that what is a cancer, how it is going to affect our body, and then what are the situations, and then how we can change our own style. Now, when you look about the cancer here, it is a one of the leading cause of death of 10 million deaths in 2020. So this is reported already that it is huge death rate as compared to when you see that other cases, and also rising 70% over the next decades. So we may be looking about the cancer is randomly increasing, but when you look about that, currently we are looking about the COVID-19. So we can see that death rate is every day, and then we may be looking at the cases are rising, decreasing like that. But here the cancer is not like that. It will be enhancing slowly, slowly into the body, and then later on there will be the cases maybe identified. It may be a third or the fourth stage of the patient because we are unable to identify the initial stage of the cancer, and only minute symptoms can be observed in the patients. So that is the problem to identify these cancer drugs and the cancer patient. Now, currently we have some of the biomarkers have been identified so that we can identify some of the cancer types. So it is also increasing the economic impact of the cancer significant, and it is increasing much when you look about the other diseases. Now here we can also see that it is 70% of death rate. So here we can see that the 70% of cases may be increased in the next 2 decades, and the 70% of death from the cancer in low and middle income countries. So here we can see that not only low, middle, even high income also we can see this death rate also observing. But when you look about the South America and then North America also we have the cancer patients. And here, when you consider about the estimated cancer incidences worldwide as men, when you look about this is a recent last year data that we can see that the highest greater than is as per it is 1 lakh population is 298.4. So you can see that is completely the North America is occupied maximum the cancer patients area. Then when you see that when you compare with the India is very less as comparison, but still we have to be ready with the caring of the patients. And in the same way that when you see that the women consideration also that it is higher in case of a North America, and then even it is almost equal to the Australia, and very less when compared to the women in India. But when you compare it with the continent or the country, it is not a same thing. But that is the only the cases when you look about the mortality rate or the death rate of the men can also be compared here. So here we can see that the maximum cases throughout in near to the China and as well. But here we can see the treatment is okay, fine. So there is no much death rate in case of North India and a little bit of South America, North American South Africa. And here you can see that in the Africa region also later some regions having higher death rate here. In the same way, the women category, when you compare both the world maps together, treatment is okay fine in the women, and then still you can see that near to this area we have the maximum death rate in the women due to various cancers. So this is just statistics of the cancer and why you have to study on the cancer. Next. So for every cell in our body, it has its own cycle. So we cannot see that a cell which may be originated from our mother, it may not be not available in our body today. But the cell division occurred such a way that the cells are growing in our body, and then our body also going to increase as per the cell growth. And in normal condition, we have the two types of the cell division. One is the mitosis and then meiosis. So here, just assume that the normal cell cycle. So we have maybe having a mitosis. So I mentioned here the mitosis. So in case the completely the cell may undergo four phases. So here we can say that the G1 phase, synthesis phase, in the G2 phase, and then mitotic phase. So this is the phase how the cell is going to be done here. So in the G1 phase, the cell may be having a chromosome, and here also is the chromosome. And here we can see that there is a double the number here. We can see the double has been done, and then later on in the mitotic phase that they may be going to divide the cells. So this is a general cell cycle. But this cell cycle will be maintained and then regulated by various checkpoints. So when you look over the checkpoint at the G1 phase, we will be having CDK4 and then CDK6. So these are the enzymes which can be called the cyclin dependent kinases. So these are the enzymes will bind with the CDKs and then it will initiate the cell cycle process. So here we will be having a checkpoint. So here we can say that these are the checkpoints. So here the checkpoint between the G1 and then S phase we can call it is a G1S checkpoint criteria. So what it will be going to do, it will find that whether cell size is sufficient, whether able to have a sufficient nutrients, and then if is there any DNA damage, and then growth factors are available to go ahead for the next cycle or next phase. If there is any problem in this phase, then it won't go. But in case of if cell cycle fails in this stage, then obviously the cell will be initiating its synthesis and then go ahead further cell division. So in case of cancer, if this phase is failed, then obviously we can see the rapid proliferation of these cells and which may be converting into cancer cells. Okay, so that's why here in our research work we are targeting to this phase as a G1 and S phase and in between the cyclin D and then cyclin E. So we took these two proteins for the targeting. Okay, fine. Remaining will be as such as a synthetic phase. After this, there will be DNA damage, and so all this will be again the criteria check whether you are able to this cell go ahead with the next phase. Now, so what exactly happen in the cell cycle? So these enzymes will be going to increase its concentration. That means the quantity may be increased in the cancer cells. And here we can see from the G1 phase 2 up to this, the cyclin, G1 cyclin will be available throughout here. Okay. And then G1 and then S cyclin, nothing but it will be cyclin D is available that will be available between the G1 and the S phase so that it will go ahead for the next phase. So in the same with the synthetic cyclins, so it will be available at the synthetic phase, nothing but the G2 phase near to this. So it will be initiating from the synthetic phase, the concentration is going to increase and then it will end at the M phase. So these are the some of the enzymes which may be regulating cell cycle. Now when you look about the difference between the cancer cells and the normal cells. So here we can see the normal cells will be able to divide systematically, normal cells. But when you look about the cancerous cells, we can see that many cells that continue to grow and then divide. So here there is no proper distinguish between these cells attachment. And variations in the size and the shape we can see, and the nucleus is completely going to be degraded such a way that the clusters of the cell without a boundary may be generated. So here we can see that the cell membrane is clearly shown in normal cell, but in case of cancer cells, we are unable to see the complete cell membrane. So here we can see that even the nucleus may be completely distributed randomly, and you can see that there may be a vacuole just like space. And here the cell is not clear with any cell membrane. You can see that the proteins may be coming out or it may be showing the completely rough surface. Do this one. So just we have discussed about the cancer incidences and cancer type and the cell cycle. Now we will go ahead the cyclin D1 and cyclin E. These are the major proteins which may be able to regulate this cancer cell, or if let assume that we are going to inhibit such type of a cell which may be going ahead with the proliferation so that we can stop this cancer. So that is a major concept in this slide. So we will go ahead one by one. Now, once we can see that there is a compound which may be extracted from the plant, it is a degulin, a novel tumorigenic agent that targeting apoptosis, cell cycle arrest, and then anti-angiogenesis for cancer chemoprevention. In the review, it has been published in 2013 by Eng et al. And Wang et al. Has been done this review such a way that he mentioned that there is a difference, maybe compound is a flavonoid family produced from the various plants. So here you can see the dairies, triflo, and these are all the completely as a leguminous plants. So this plant having a degulin, which may be as a anti-tumorigenic agent, targeting these three cell signaling pathways. So we will see those now. So for our research work, this is the basic a paper which has been taken further. Now here we can see that the degulin can bind on various pathways. One is the cell cycle arrest, apoptic pathway proteins, and then anti-angiogenesis pathway, where we can try to inhibit these cancer cells. So here it may be initiating, if we just assume that the degulin can able to inhibit the cyclin E and the cyclin D so that it can stop the cell cycle, so that we can stop the cancer cell growth. In the same way, apoptosis, and then say way that anti-angiogenesis. So here we can see that where we can find there is a symbol like this inhibition, so that it can be able to inhibit particular proteins. So this is the basic concept how you can proceed in research work to find out from the basic level to higher level. So all these proteins considered as a target proteins, but we can see that which protein can be inhibited so that we can stop the pathway. Now we will come to that. Is it visible to everyone?
Yes, yes.
Yes, sir,
Yes.
it's visible.
Okay. Okay, fine. Okay, fine. Now you can see that the chemical compound actually that is called as a degulin, which is existing in all these leguminous plants. So this may be available around to us, but we don't know that it may be treating the cancer diseases. So until unless we do some practicals and then experiments. So how we can proceed into this? So what is the importance of this degulin, which has been reported by many number of scientists? So here we can see that in 2021, so recent publication shows that the anti-cancer activity of degulin is a HER2 positive breast cancer. So here we can see that it is able to inhibit the cancer, and then degulin cured the colorectal cancer in 2013, and then induced apoptosis by targeting the EGFR, again epithelial growth factor in this AKT and then IGF-1 AKT pathways, and then downstream regulation of apoptosis and antigenesis cell cycle arrest. So we can see that various importance in our treatment. So this is a major molecule which is able to treat many number of cancer type diseases. But we have some problem. Why degulin is not a drug? So why company are not developing this molecule is very important when you do in the animal cell lines, but when you start transferring this molecule into a clinical trials, it's not success. So here it is reported as a Wang et al. So you can see that 2008. So to show that if you change that there is a 7A alpha carbon, then it may be able to treat, or it can be given for as a drug molecule for the patient. So here we can see that the decomposition is okay, fine. So it is able to once enter into the patient sample, that will be able. But here we can see that the stability and then biological activity. So here it shows that the in case of this stability decomposition, so it is biological activity and then stability. When you look into this, so we can find there is a little bit of change in this one. So even you can see that there is a toxicity level maybe maximum in our body. So to avoid these toxicity levels, so we have changed, modified this molecule in different ways. So just we will show that. And our methodology is such a way that we have retrieved this degulin compound chemical structure from the PubChem database. Then we have done some statistical analysis and then filters by 95% similarity of the PubChem with the rule of 5, nothing but if you look about that hydrogen bond donor, hydrogen bond acceptor. So, and as well, the log p value should be less than 5, and then we implement the Lipin's rule of 5, so that molecular weight should be less than 500 Daltons. So, like that. Then we have taken these many compounds, then we have done some energy minimization methods, so we can able to do on computer. Next, here the cyclin D1 and then cyclin E crystal structures are already available in the protein data bank. So we have downloaded those two structures and then we'll start analyzing the active binding site. So here we can see that main binding site where we can the cyclin can able to bind to this. So these are the compound which may be showing that some of the amino acids in the active site where it can able to bind with the these are the cyclin, and then cyclin dependent kinase can able to bind here. So as well the compound can also bind in this phase, so that isoleucine and then lysine, arginine, lysine, and asparagine 145 and then threonine 177. In the same way that some of the amino acids may be seen according to the main backbone chain of the protein, it is asparagine 2190. So these numbers indicate the amino acid number. So amino acids will be the length of the chain. So when it is folded, then we'll be having a complete these amino acids. So in our body, whatever you are taking inside the external molecules, that will always bind with the proteins. So then once the energy may be transferred from that molecule to the enzyme, then we can say it is a biochemical reaction. So it is happening in our body, so that we can call it as a biochemical reaction. So here, whatever we are going to design the molecule, first we have to find out where is the cavity, where it is going to bind. So that is nothing but the prediction or identification of the binding site. Now here we can see that we have done the various calculations based upon the docking method. So you can also find that there is a grid box, nothing but the box may be generated in the Autodoc software, and we can predict that you can also say that yes, this is the molecule, the degulin can or derivatives can bind into this cavity, and later on we can see that what are the interactions between these amino acids. So that is called as a docking, and by forcefully we are allow this molecule to bind into this molecule to see that how much energy may be achieved or interactions can be calculated. And later on, we're going ahead with the simulation methods in a computer to find out the stability of these molecules for the time being. So here we are just listed the top 4 molecule here. So number 1 is the degulin. So here we can see that the binding affinity minus 8.9 kilocalories per mole. This is the energy of the affinity that is binding to this. But when you look about the degulin 32, the molecule is having a minus 10.2. So it is a lower the value. So if you take the number line, then we can see the minus 8, minus 9, minus 10. That means it will go into down. So when you look at the lowest binding of the interaction, that can be shown as a best binding molecule. So you have to remember the concept clear that it able to bind it properly rather than degulin to degulin 32 molecule. So the molecule I am going to reveal that what is the structure that are been changes done in the main degulin molecule, so that why we are going to achieve this binding affinity molecule. Even in this case, the interacting residues also when you compare the previously the degulin is 35, lysine 35, but here you can see that it is also interacting with more number of amino acids, nothing but you're able to uh, stay longer period as compared to the degulin. And here remaining the bond type is again hydrogen bonds, and then interaction, the distances that I'm going to show in further slides. Here we can also see that it's related to the cyclin D1 receptor in the same way that we have done the same compounds already interacted with the cyclin D1 with the stably conformation as a receptor here. Now here we can see that the binding affinity of the degulin and its derivatives with the crystal structure of cyclin E1, and here we also its derivative, the dynamically stable conformation of the cyclin E receptor. So there are two things are there. So if you see the comparison, so one is the crystal structure, nothing but later in the crystal structure is binding. So the crystal structure, when you look about that taking this conformation into the simulations, then I want to see that how it can bind into our biological system. So we have simulated the molecule, and then we have seen that how the interaction may going to change again. So here, this one we can see the cyclin D1 is the binding mode of a degulin with the crystal structure of the cyclin D1. And here we can see the comparison that it is interacting with the only lysine 35. You can see here the previous reported information. But even in the same molecule, when it is binding with this modification of the molecule, it is able to bind with the many number of amino acids. So previously I've shown those amino acids as aspartic 97, valine 96, and then lysine 35 amino acid, and then these amino acids. So like this, we can able to identify that how why degulin is not as a drug here, and then why the modified degulin can able to be a drug molecule. So here we have shown that all the dashed lines as black, it indicates the hydrogen bonding. Okay. So, but in reality or the native state, that we can't see that any ribbons or any sticks on a model in our body. So this is the representation of the bones and as well the angles and then atoms for our understanding carefully, so that we have taken these images. So when you say that the protein and the ligand RMSD, the RMSD in the root means square deviation. So here that how much stable a ligand when it is bind with the cavity. So let's assume that if it is existing in the cytoplasm, then it is moving further. So how much time it will be stable in a cavity, that can be identified based upon this root mean square deviation of the backbone of a protein. So if it is changing the movement in the cytoplasm itself has a liquid state. So it is completely that how much it will be stable in the cavity of a protein, that can be seen here. We can see that the red line indicates the ligand fit protein. So nothing but how much it is fitting. So initially it is unstable, later it is become as stable. And then here we can see that it is C alpha protein, nothing but completely the backbone of a protein also stable up to 100 nanoseconds of a time period when you have done in a computer. So in the same way that it is also verified with the degulin with the cyclin E. Previously we have done for the cyclin D. Now when you are doing with the cyclin E also, we can see that there is a little bit of a disturbance, nothing but you can see there is a 16 nanoseconds of a duration. Okay, just I'm keeping a molecule and then I want to check it out for the 5 minutes. Okay, then normal time we can say it is a 5 minutes. But we want to run for the complete 100 nanoseconds of a duration and to see that where will be going to be stable in our body. So in a normal sense, we can say that if let us assume when you're taking a tablet inside, so that in the tablet there will be a generic or a very important molecule which may be going to treat your disease, will be going to bind with some other protein. And later on, after some time of a interaction, later on this molecule will be going out from the cavity and then it may undergo degradation or it may be released from your body. So it may be through the urine or it may be the sweat or the feces. So every molecule when you're taking the outside, it has to remove from our body once the energy has been transferred in such a way that. So in the same way that we can find these interactions carefully understood, that during the time period, the movement can be observed as a protein RMSD as a y-axis and x-axis you can able to take as again number of seconds, or it can be called as a nanoseconds of a duration. It is a time very small. Okay, so within a fraction of second we can able to get a new cell, but if I want to observe completely, then we have to increase the time. So in another way that we can able to take the example, so most of the time in a cricket, that once the ball may be hit by the batsman and it will be going ahead and then somebody took the ball. So we can say the catch. So sometimes if there is a dilemma there, then we can call it third umpire. So whatever happened, the incident, it will be observed. It will maybe a longer time. It may be 5 minutes or the 2 minutes, and then you can give the decision that whether it has been done properly or not. So nothing but whatever happened incident, when you increase the time period, then we can say it is a you are trying to analyze the interaction. So in the same way that here, whatever happens in a one fraction of a second, now we're increasing the time for the nanosecond to observe at microscopic level. So this can be called as a microscopic level. Nothing but you are going depth into the protein ligand interactions. Whatever happening within a fraction of a second, we want to increase the time to observe that we are going to the minus level of 10 to the power of minus 9, 10 to the power of minus 12 as a second. So we can call it as a nanosecond or the picoseconds. Okay, this is observation as a protein and the ligand. So when you see the simulations, we can see the isolation 161 amino acid fluctuation maximum. So it means to say that it is trying to interact with a compound maximum time and then able to stabilize this new design molecule in the cyclin D1 molecule. So here we can say that the maximum fluctuations in this case, so that we can call it as a 100 nanoseconds of duration. Now here we can see that when look about the protein between the two terminal, one we can call it as N terminal and the C terminal. So in between it is completely stable, but here we can see there is a change, complete fluctuation in the C terminal and as well as the N terminal. It indicates that there is a completely freedom for the N terminal and C terminal. It is a completely as a flanking chains. You can call it as a flanking chains where there is no any bond or it's not folded properly at the C terminal and the N terminal so that they are free. Whenever it is simulating in a cytoplasm, they are able to move freely and then fluctuations. So here we can able to find out the interaction stability between the free energy calculations for the cyclin D1 and then degulin, and then as well the cyclin D1 and then degulin, which is minus 77.79 and minus 38.80 for various molecular mechanics calculations, or we can also call it as a generalized bond and then surface area quantum solvation. So based on this complete data, we have done the research work and then we have published into these scientific reports in 2019, and it is having a very high impact journal. And then later on the same work previously also we have presented in the Paris about the in the Journal of the Proteomics and then Molecular Medicine as an international conference here. And one of my student, PhD students, has attended the conference by Mr. Kiran Bharat Lokande. He has done the this complete work that I'm presenting here. So later on we have done some other using computational chemistry, and later on what I have done the research work, then we come to know that yes, there is a requirement of the change in the modifications. So here we can say that the complete molecule that has been changed at the B, we have divided into A, B, C, D chain, completely rings. So here when you do the changes, the truncation thing, but if you remove this chain, then or the B and the C, then we are able to get such type of molecule. So R1 or R3 may be any functional group that may be added into this molecule to get the new molecule. That's thing, but we can call it as a drug design properties. Okay, so we are going to design a molecule from the knowledge. In the same way that here ring truncation of the B, if I want to make the breakdown here so that you can get the new molecule, you can see the benzene with the R2, R3, and then R1 like this. So you can see the difference between the degulin when you see there is a C, when you remove the C, and then just replacement of the C with the nitrogen. So here, just you can see that instead of the carbon, we are replacing nitrogen, then we get the new compounds. Then we have seen that how many compounds are going to get by these modifications. So here by 1843, and R2 by this we can also get 1843 molecules, and in total 3686 molecules have been generated. In the same way that by various modifications we are able to get almost a huge amount of 11,058 molecules have been generated by the computational chemistry methods. Then we have proceed with the filtration methods, and we got after filtering we got the 9,930 drug-like compounds have been got from this method by the software called as Schrodinger. And here the cyclin binding pattern already defined, identified, and then we have taken these binding sites. Then we proceed with the grid box generation the same way that what we had done the previously. Then later we found that there is two types of the docking methods. What is the specific precision method, and then there is the extra precision method as both, and then we identify this data. Next, we further mapped these energies as a minus 5.674 kilocalories, and then cycling with the again docking method minus 7.354, again minus 7.8548. Again, the cyclin D interaction molecule is a mapping with the specific amino acid. You can see that how the molecule is able to bind. This is the cavity. The surface can be shown like this, and the molecule may be able to fit into the cavity. So there are one thing which is a 3D, it is the 2D structure where we can try to understand the interaction of these amino acids. So in the same way for the cyclin E, the presentation, all the amino acids, how they are interacting. So all these done with the software tools. So when you do the simulations in the same way for these new compounds also for the RMSD, but it has shown into cyclin D1 and then D4 as these dead molecules together, and all of them are able to stable into the molecule. So RMSF, previously we have obtained the isoleucine, but here we can see that 241 amino acid is fluctuating maximum, so that we can say that whatever taken the molecule is the a pink in color, or we can say that this yellow color. Can we see the cyclin D1 and then this cyclin dependent kinase 4 when it is binding, so we can see the maximum fluctuation in this case. So these are the complete simulation results from these studies. So based upon these results, we come to a conclusion that is PubChem database having a very good potent molecule, but just we require to screen the molecule by this computational technology. And initially we got very less molecule. Later on we found that 181 derivatives dragged out from these PubChem databases. The potential and the efficiency of molecules may be screened much more here as a cyclin D1 and another molecules. And apart from all these molecules, it has been defined that the deglin 32 and then deglin 40 and then deglin 49. So we have given our own name for in our lab. So these molecules consider as a very good potent inhibitor as compared to other molecules. So these are the conclusions from this. So already mentioned about the T2, the molecule that what I have shown the comparison with the energy calculations. So we reported that these molecules may be taken ahead with the clinical trials. So these are the various mathematical calculation and then docking interaction data now currently. So based upon this data that I presented this work in Paris in 2017, and he is Mr. Kiran Bharat Lokande who worked completely on this project. And this project was sponsored by the DST, and he also got very good research excellence award 2020 for Kiran Bharat Lokande. He also participated in drug discovery hackathon, and then he got cash prize of 25,000 as in the first phase. And I would like to give the acknowledgements to my head of the institution, Professor Renuvas, and then this work carried out by the Kiran Bharat Lokande. He is currently is a PhD student under me and senior research fellow from ICMR. Previously also worked with the DST project, so it is a part of the DST project, and as well as the ICMR funded PhD project. And we have completed in the DY Patil Vidyapeeth this project, who helped me in the Professor Neelu Madam and then Professor Jacob, who is the director and the research coordinator. We have used the software as a Schrodinger software, who has given the license to use to calculations, and then who helped about the Bioinformatics Center, Savitri Bali Pule University in Pune. They helped us, their software they have given for us calculations of MMGBA calculations. So I would like to thank all of them for their constant support in my research group, and thanks to everyone. And any questions, so I welcome all of you.
Hello, good morning, sir.
Good morning.
Hello, am I audible to you, sir?
Yes, audible.
Actually, sir, I work on the breast disease data set and also for heart disease data set.
Okay.
Hello. Hello,
Yes, sir, I'm able to listen.
sir. I want to publish a paper on. Yeah, I want to publish a paper on that. Can you give me some brief discussion on that? How to publish a paper on breast cancer with constant data set and heart disease data set for that?
Okay, fine. So whether you have discovered
I use
you
machine learning, deep learning algorithm.
yes, so there are various research journals are available to publish your data set which has been already available publicly, and then you want to do some innovation and then new things from the deep learning and then machine learning. Is it correct?
Yes, yes, sir.
Yes. So
I
we have
use CNN for that.
yes. So first you have to search this data
Hello?
whether it is yes. Are you able to listen, Divasa?
I actually, yeah, sir. I used Kaggle dataset, but I
Hmm.
want to take my own dataset from hospital like that,
Okay.
so that I can modify my program according to that.
Yes.
Hello?
Yes. Yes.
Hello, sir. Am I audible?
Yes, sir. Audible, sir.
Yes, sir. Can you give me brief? Yes, yes. Just
Okay.
suggest me how to publish a paper on that, sir.
Okay. First you have to go for elsewhere journal. and then search, you type the AI and deep learning
Okay, sir.
method in medical imaging, or if you're taking the data set, which type of data set you want to take, whether the data set of the cancer images you want to take, or CTSC, or which type of data you want to work.
Okay,
Is
sir.
it okay? So then you can
Okay,
search
okay, sir.
the journal, which is basically called, it is a, we have the journal of the computational biology is one of the best journal if you want to publish the research type of AI and then deep learning method.
Okay.
Okay, fine.
Sir, do you have MRI data set by your own? Can you share the MRI
No,
data set to me?
no, MRI
MRI
data
scan
set,
is there now?
MRI scan is available on the National Institute of Cancer on NIC website. You can able to download.
Okay, okay, sir.
Okay, fine. Okay, any further questions on the topic?
Okay, now we are moving to question answer session.
Okay,
Sir, we have some creative questions from the audience. So I will call up the names of people who have got their question so that they can interact.
okay,
Okay.
ma'am.
Our first question is, what tool was used for modification in decoupling?
What is the?
What was the tool used in modification in decoupling?
Okay, so we have used the Schrodinger software to modify this molecule, and even we have used our own knowledge-based technology where we can find out the new drug molecules. So in a Schrodinger software, we have the provision to
Hello,
do.
sir. Is my voice audible?
Yes, yes. So we have used the Autodoc and then Schrodinger software to design these new molecules.
Hello.
Hi, yes, ma'am.
Yeah. And their question is why decoupling can't be used directly as a talk. Yeah.
Okay, so I already mentioned that the degulin having a maximum toxicity as compared to the other compounds. That's why we are not using a degulin as a directed drug. So we want to modify the drug molecule from this degulin to other type of a molecules. So at a higher doses, it is going to be converted into toxic to our body. That's why we cannot able to take this degulin as a drug.
If anyone having any question, you can unmute yourself and ask.
Good morning, sir. Hello, sir. Am I
Good
audible?
morning. Yes, audible.
Sir, I had one question. Like for publishing in a good journal, approximately how much nanosecond simulation is enough? Like 40 or 50?
So first you have to run for 100 nanosecond duration, and then you have to proceed for the 500, then you proceed with the 1000. So here we can able to identify that whether it will be stable
Right.
for the 100 nanoseconds. If I am going ahead with the publications, they are going to ask for the up to 100 nanoseconds.
Okay, okay.
So
Right, sir.
even further, sometime if you have a very good computational resources, then you can go ahead up to 1000 nanoseconds of duration. But make sure that the protein is stable in your GROMAC software or any other software.
Right, sir. Gromax. And
Okay.
sir, one more thing for the modification of this, what software was used, like Chem Axon or something?
So this is actually 3D structural modifications. So this can be done by the Schrodinger software.
Schrodinger suit only.
Yes, yes, Schrodinger shooter can be used for these modifications. But if you want to do the knowledge base, then you can able to the chemaxon can be taken and then you modify the compounds according to your knowledge. Later in the
Oh,
as I have done the breakup at the B chain, so you can able to break up
right,
and then you can design your own molecular data set.
right, sir, right, sir. And sir,
Okay.
about the MMGB assay, was Schrodinger used for that?
Schrodinger will going to provide this MMGBA calculations, but let's assume that if you don't have a commercial software or we don't
Right.
have a resource of this one, then you can able to in the GROMAX itself.
Okay.
There is a separate software is available MMGPA calculations which already designed and then launched in 2019 on the GROMAX site itself. So you can download the module which is basically the Python module.py. Okay, so you can run the program by complex
Right.
structure. By taking a
Right.
complex protein ligand complex can be taken and then you can able to run in the GROMAX. Then
Right.
you will get these values.
Please. Great,
Fine.
sir. Thank you, sir.
Thank you.
Yes, sir. Good morning. So I have a question. So before that, sir, your talk was great, and this is a very new kind of a thing that we are learning that degulin also has some anti-cancer property we can get out of it.
Yes.
Sir, but I had a question, like very technical one, like you have used which software for this docking purpose?
Okay, so initially we have used the Autodoc software to design a grid center, other things. And later on we move ahead with the Schrodinger software. We will have the automated method where you can do these calculations.
Okay.
Okay, so
So
let's assume that if you don't have a commercial this one with a Schrodinger, then you can able to do with the Autodoc Vina directly, where you can do almost a thousands of a molecule at a time. You can dock into the grid center.
but Autodoc has like one by one we have to do all the
No,
docking
no. You can able to do almost all 10,000 to 50,000 molecule at a time. Autodoc Vina.
in
So
Autodoc.
yes, there is a script also available online.
Okay.
So even you can get the script, and then if you run the script where you can mention the protein name and then ligands completely, you can put into one folder. Okay. Then all the ligands may be converted into PDB to PDB-QT by the Open Babel software. Okay, so you will get all the 10,000 ligands at time, and then you can run the program on your command line. Even you can run on the Windows, or if you want, you can run on the Linux based.
And
Okay, so yes.
specifically in Degulin, what was the docking score in Autodoc in the beginning?
So it is giving similar minus 7.5
Minus 7.5. Okay.
for
And
the Degulin, and then when you look about the Degulin, which is modified, we are getting minus 10 points. So there will be a lot of difference between both of them.
okay, so what modification that you have brought in this new molecule that you have made so that it is less toxic?
Okay, because as per the observation, the Wang et al. He mentioned that if we do the changes at the B and then C and the D chains, then it will may be probable to get a good molecule. So that is his prediction. It is
Okay,
based upon
so
his wet laboratory.
yeah, so you have done this modeling in any like any trial in any animal models or
No, not yet, not yet. The project was only for the computational thing. So
okay,
we have
so
done
it
only
is
for the
proof of
computational analysis. Yes.
okay,
So
okay,
further, we would like to take ahead in the clinical trial. So later on, we will see that if funds are available, then we can able move ahead this molecule into clinical trials.
okay, fine. Thank you very much.
Yes.
Sir, may I ask one more thing?
Yes, yes, proceed.
Listen, sir, you've told that around like 11,000 molecules were there after the modifications.
Hmm.
So for screening, I mean for docking, after the docking and all, only 24 and 5, like 24 for cyclin D, I
Hmm.
think, and 5 for cyclin E were there. So for all these, can Pyrex be used, open based software, or we
Yes,
have to
yes.
use commercial?
No, no, you can use the free softwares. There is no problem, but you should have a very good server where you can run all these calculations. It cannot be done as
Oh,
it is on a laptop because it has to run almost a 2 to 3 days or the 5 days time duration. Continuously it has
right,
to
right,
run without interruptions. Okay. And then we
right.
have done this completely on a GPU optimized supercomputer where we can able to get the result as well as possible.
Okay, okay, sir. And sir,
Okay.
may I ask, in what supercomputer have you done all this? Like
So
in
this
India?
these calculations done the GPU based servers.
Okay, okay, sir.
Okay,
Okay,
graphical processing unit servers are available currently in the market. So it may be come to the 5 lakhs to up to 10 lakhs, and it is beyond this number of requirements. And later in that, if you don't have a server like this, so you can able to take the IIT Delhi supercomputer can also be available currently.
Right,
So
right, right.
you can use that IIT Delhi supercomputers for you these calculations. Even you can also require the CDAC New Delhi, Pune also having a supercomputer, so you can use that servers also. So that you can able to create a username
Right, right,
password
right.
and then you have can able to run these calculations. Okay.
Right, right.
And
Yes, sir.
these
Actually,
software can
yes.
also work on the Google Cloud also. Google Cloud,
Google Cloud
Google
also, sir.
Cloud, yes, yes, yes. So you can also register. Already you have the Gmail, then you able to log into Google Cloud, and then you can load, upload, and the things can be done. You can run the Vina also there.
Right, sir. Actually, sir, I had participated in this DDH
Okay.
one, but I
Yes, yes,
wasn't able to do the molecular simulations because the crowd was so much in the server.
yes, yes.
I
CDAC
think,
server.
CDEC Pune, I think
Yes, yes,
Param
yes, yes,
Shrestha
yes.
one. So the
Yes.
crowd was so much, so that's why we weren't able to do the molecular simulations of all of these.
Yes, whatever you gain the experience in the drug discovery hackathon, it will help you to do the same setup in the IIT Delhi.
Right, sir. Right.
Okay, IIT Delhi supercomputer is free for the academics, and then sometimes they may charge a little bit of amount for your number of hours. Okay.
Okay.
Okay, there
Okay.
are
Okay.
two servers are there. One is the IIT Delhi chemistry department having this one, and then main also having another IIT Delhi main. Okay, so
Okay.
you
Okay,
can
sir.
use those
Okay, sir.
resources if you want.
Right.
And if you're able to do some the MMGBSA on the Schrodinger software, then you can also approach Pune University for your titulation.
Right, sir. Thank you, sir.
Thank you, ma'am. Okay, further any questions regarding the bioinformatics and the applications?
Hello.
Yes,
Yeah.
ma'am.
If you
Rajendra.
if you have more if you have more questions, you can send those questions to our social media handles and email ID. Okay. Thank you very much, Professor. The talk was informative and enlightening. We learned a lot. Thanks for taking out some time from your busy schedule for catching the vacation. Now, I will request to Anthony to feed the feedback from will share soon. Okay. Now I invite Mr. Mohammed Sahil to make one important announcement and present a vote of thanks. Sahil, please.
Yeah, thank you, Rosalini. Hello, everyone. Myself, Muhammad Sahil. I hope everyone is doing well and fine. The session was very insightful, and I hope you have enjoyed a lot in learning these new stuffs in the field of bioscience. As this event was very insightful to learn, there are many such more events in the field of bioscience society. Okay, Bioscience Society is going to conduct in future to promote the biological research in young minds, uh, to give the freedom to the research ideology. With the Bioscience Society, extend our hands for the students and research professionals outside the NITR chapter to join us in our mission and contribute the betterment of the society by being the affiliate member of the Bioscience Society NIT Rourkela chapter. So it's an amazing opportunity for the people from other school, colleges, different domains, etc., to come together and meet in a single platform to release the wonders of bioengineering and biosciences. So buckle up, guys, and stay tuned with us in the WhatsApp group if you want to bring some change in the biology and bioengineering research and development. Fill the form. We'll soon drop the link in the WhatsApp group. Okay, which we'll be sharing after the feedback form in WhatsApp group. For more information, you can contact us, any of the group admin. Okay. Now, finally, on the behalf of the Bioscience Society, I extend my sincerest thanks to everyone who has joined us today, and thank you very much, Professor. We are really very grateful to have you on board with us today's morning. And last but not the least, a big thanks to members of the Bioscience Society who have worked very hard for making this event successful one. And thank you, everyone. Thank you. Hope you enjoyed a lot. Have a nice day. Thank you.
Okay, thank you, Mohammed. Okay, there is a query from the Sai, and then last latest query is there. Okay, Sai Megan is asking, sir, will we answer my query, and can we go for a process in which we can differentiate the cells near the tumor cells like plants? Okay, differentiate the cells near the tumor cells like the plants. Go for some modifications in cells that contain oncogenes. So currently, this type of a technology is still it is under trial method. Actually, we can call it CRISPR technology, or we can say that the cells can be differentiated between the cancer cells and the normal cells. So in the cancer cells, even you can touch it, the tumors, it is very hot, but the normal cells may not be hot. So anyway, we can able to identify that near to the cells or the cancer cells or the normal cells. So this is we are using the chemotherapy. Sometimes we will be giving it drugs directly to the patient to take from the esophagus, but sometimes we can give the drug directly to the tumor itself. So we can call it a tumor targeted drugs. So in that case, we are going to kill the cancers. But when you see that, go for some modification cells that contain oncogenes. So this is very important when you look at the oncogenes going ahead to release these problematic things in our body. So here the oncogenes cannot be modified at the higher stage. So once it is expressed, the protein is already available in the cytoplasm, and then the cell cycle already altered its one function. That's why it is too difficult to change the oncogenes at the initial stage right now. And that's why just we are going ahead with the different genomic technologies and then engineering methodologies. But once the cell already done its modification, so there only one thing is that whether you can go for radiotherapy or the chemotherapy and then other methods to treat it. Okay, so thank you one and all for giving. Sai, I think you are satisfied, or any further questions? So here there is a de-differentiation is nothing but how can you differentiate these cells completely into normal cells into tumor cells. Okay, that is the major thing. Okay, thank you one and all, and we'll meet
Yeah.
again. And you can also visit our site and then our labs on the main website if you want to visit the lab other things. Okay, thank you.
Thank you,
Okay,
sir.
thank you, sir.
We have
Thank
shared
you.
our feedback.
We'll drop the feedback form,
We have
guys, in
shared
the
our
chat
feedback.
box. Okay, so and guys, for affiliate program, uh, don't forget to fill it. We'll soon drop the link as well of the affiliate program. Okay, in the WhatsApp group, or in case of any doubt, convey the same in the group, or you can also DM any of the group admin. Okay, thank you, guys. It's time to wrap it up.